All right. Hello, everyone. Welcome to our recorded lecture beginning chapter 5, which is on sensation and perception. So, sensation is going to be the process of taking information that's out in the world, whatever that information might be, and getting it into the brain in a way that it can be used. That's going to be a sort of our primary focus today. When we come back on Monday, we will talk about what the brain does with that knowledge, with that information, once it has it in there. So, beginning talking about sensation. So notice the word is very much like senses, and that's what we're talking about here. What are your senses, and how do they operate? So the first thing to note is that when it comes to research on senses, there's a variety of different levels of knowledge on each of the senses. So, the number one sense that we know the most about is vision. We have a tremendous amount of information on how vision works, how it operates, how it can be fooled. So most of what you're going to see in textbook and in our classroom time is going to be about vision, just because that's where we know most of it. Second, we know a decent amount about hearing and touch. Not as much as we know about vision, but we know the general processes, we know the brain areas involved, we know the sort of general ways we can provide illusions for both hearing and touch and how the general process works. And then last, we know the least about taste and smell. And I think if you look in your textbook, there might be a paragraph about taste and smell. In other textbooks, it maxes out at about a page on taste and smell. We just do not know very much about how those senses operate, what they're hooked to. There's a lot of information out there on taste and smell that needs to be found out. So, because we know the most about vision, that's where we're going to start, and that's where we're going to spend most of our time. We're going to spend most of our time looking at how does vision work, how does it operate, and how can we fool it. All right, so primary point about vision is that vision is, in its sort of technical term, an interpretation of electromagnetic energy emitted or reflected from objects. So there's a lot there. So let's start kind of unpacking this one by one. So number one, vision is an interpretation. And what that means is that vision has a lot to do with our psychology. There is information in this energy coming at us. However, what we say is, what we understand as vision has a lot to do with. Right, so what do we mean by electromagnetic energy? Well, electromagnetic energy is just a type of energy that exists in the world. And if you look at that diagram on the left-hand side of the slide here, you can kind of see the types of things that we would consider to be electromagnetic energy. These are things like the broadcast bands that allow you to send information through your cell phone or to television towers, how you get your television or radio, all that stuff. Radar, infrared radiation, visible light, which you can see in there, and then also some things like X-rays and gamma rays. Those are all examples of the type of electromagnetic radiation. And really the only big difference between all these forms of radiation is what the wavelength of the wave is. So electromagnetic energy is a type of energy that travels in waves. That's all we are going to care to know about electromagnetic energy, right? We're not going to have to get deep into the physics of electromagnetic energy, but just know that it travels in waves, as you can kind of see I have on the right side there. And the difference in electromagnetic radiation has a lot to do with how far apart the waves are. We're going to spend most of our time in the visible light spectrum from here on out, and because that's what our eyes react to. And the wavelengths we're talking about here, we're talking about 400 to 700 nanometers. And a nanometer is 1/billionth of a meter. So that's kind of the distance we are talking about. So very, very small differences, very, very minuscule differences. But they have a lot to do, or a lot to say about what it is we actually interpret when that electromagnetic radiation comes in contact with our eyes. All right. Oh, and I should mention another, the general term for electromagnetic energy is light, whatever we want to understand as light there. Okay, so let's talk about color. As we said, color has a lot to do with what wavelength of light we are experiencing. And as you can kind of see that, the longer wavelengths produce red interpretations, shorter wavelengths produce blue interpretations, and the medium ones are obviously green. And then along the whole spectrum there, you're going to see all the other colors that we typically perceive as purple and yellow and orange and all of those. Right? So it has everything to do with the distance between the peaks in the wave. That's the wavelength. Right? How far apart are those peaks in the wave? If they are relatively longer, and remember we're talking about 700 nanometers there, so still very, very small, but relatively longer wavelengths, those are red, or perceived as red. And the relatively shorter ones, at about 400 nanometers, are perceived more as blue and purple. Okay, so that's kind of the physics lesson. That's as much as physics as we need to get into, right? Just understand wavelength is important. It produces the interpretation of different colors. Okay, now let's talk about a little physiology associated with the eye. Let's do the animation here just kind of so it can be a little slower. So first off, there is a major structure in the eye that kind of has all the light responsive cells in it. That's the retina. Now the retina is on the back part of the eye, but the back half of the eyeball. And if you look in your textbook, you can find a diagram of the retina. So what's important for our understanding of the retina is that it has two light responsive cells in it, right? It has rods, and you can kind of see both of these types of cells on the top layer of that diagram that's below the bottom, right? So first we have rods. Rods are really good at sensing light and dark. That's what they're good at. They're also good for seeing in very low levels of light, and they're very good at seeing movement. However, rods cannot detect different colors. They only respond to, is there a light shining on this cell, or is there not a light shining on this cell? They don't differentiate colors at all. They don't have the ability to do that. But what they make up for in not being very good at seeing color, they allow you, this is your sort of dark vision type of things. They allow you to see movement. Rods typically are your peripheral vision, and we typically are better able to see movement out of the periphery than we are when we're looking straight at something. If something is moving very, very slightly, it's much easier to see those very, very slight movements in the periphery of your vision than it is if you're looking straight on. An also important thing to note about rods is they really stop responding if they're exposed to very bright light. So if you're outside and it's nice and sunny, your rods have basically stopped functioning. Because everything is bright and there's light on everything, and they just don't respond anymore. So if your rods are exposed to this really bright light, they're just not doing very much. All right, so that's one type of cell that's in the retina. The other type of cell is called a cone, and notice in that top layer we've got two cells that look very much like a rod and a cone. That's what those cells typically look like, and that's how they were named. We looked under a microscope and said, oh, these look like a rod, and these look like a cone. All right, cones are basically the opposite of rods. Right? Whereas rods are really good in sort of low light, but they don't do well in bright light. Cones respond really well in bright light. In fact, they respond best in bright light. They also, where rods don't help you with color, cones are your color vision sensors. You have 3 different types of cones in your eyes, all of which are responsive to different wavelengths of light, and that's how you can see color. You have a type of cone that responds to red, a type of cone that responds to green, and a type of cone that responds to blue. And the mixing of those and how they respond helps you interpret the other colors in between. So cones are how you interpret color, and you can see color vision. You can see color vision vision in color because you have cones. Cones have greater what we call acuity. And what we mean by acuity is that they are the sort of sharp vision. They're able to they allow you to see in very great detail. The amount of detail you can see when you're looking at something with a cone is much, much greater than you can see with a rod. So it's much, much better. You see in much greater detail. When you're looking very, very closely at something and you're trying to inspect it and see all the details about it, you're essentially looking at that object with entirely cones. And last but not least, it's important to note that rods and cones are distributed differently in the retina. Cones you're going to find primarily in a place called the fovea, whereas the rods are much more about sort of the portions of the retina that correspond to your peripheral vision. Your cones are the fovea, and the fovea is your focus, like the point of focus of your eyes. And that's where you will find mostly cones, and that's why you have this greater acuity, right? You want these sort of things that are really good at responding in bright light, seeing color, and can see in great detail. You want all of those in sort of one particular place, right at that point of attention. And when be in mind wherever you're moving your eyes around to, that's where your kind of fovea is pointed. So wherever your focus of attention is, that's where the fovea is. All right, so you can see this diagram here. Right? We've got the fovea. You can see it's kind of pointed right on the back of the eye. It's this tiny little depression in the back of the retina that allows you to pack more and more cells on that same surface area. So that's how you get some of that larger acuity going on in the fovea as opposed to somewhere else. There's just a load of cells packed in there as densely as you can get them, and they're all responding to the light that's falling on that particular part of the retina. And you can kind of see the whole process here as you get a ray of light that comes from the outside, comes into the eye, gets changed around by the lens. You're going to want to check out the textbook for all of this stuff. It gets moved to the back of the eye where the retina is, and then the image eventually falls in the fovea. So that's how all this works. So the fovea is, as we kind of pointed out here, it's the area of the retina with the greatest visual acuity, which means the greatest detail in the vision. You can see the most details, the most small differences in the image when you see something at the fovea. And this is a very densely packed area, mostly of cones. There are not very many rods in the fovea at all. There are a few, but not very many. All right. One final thing to note about the anatomy of the eye is that you do have a blind spot. So if you look on this diagram here, you can see that the optic nerve exits out of the back of the eye, kind of right in the middle of the retina. And so where that optic nerve exits, there are no receptors. There are no cells that allow you to respond to the light that falls on that spot. So your brain just is not receiving any information from the point of the retina where the optic nerve exits the eye. So there are no rods or cones there, and so you cannot see anything there. There is no information. However, we don't notice this blind spot, right? We don't pay attention to it. And the reason is because our brain kind of fills in the information. So if you want to try to find your blind spot, what you do is you want to hold your finger out and you want to pay attention to something like really focused in front of you, but keep pay attention to your finger where it is, but you got to keep your eyes looking straight ahead. And you're looking to move your finger about 30 degrees away from center. Like if you're way out here, you're not going to find it. It's not over here. Right? It's going to be closer to the point of fixation and just below the point. And if you do that, and you can kind of pay attention to your finger, you'll kind of notice your finger just disappearing. This is where mine is right here. And your finger will just disappear. And it'll look either smaller, or you'll see like the white piece or I see a white screen behind it, because my brain is filling in the blind spot with whatever happens to be around it at that time. That's something your brain is really good at in perception. It's just filling in these spots that it doesn't have any information for.